Whenever you want to start.
Okay, so today, myself and my partner Emily are going to be presenting on a paper that goes in depth into the conformational flexibility and the mechanism of something, what's known as a adenine ribose switch. In particular, we'll be looking at specific domains within this RNA ribose switch and different states, and really what those states configure to in different environments, and what we believe the overall mechanism looks like. So, you want to hit the next slide? A little bit of background. A ribose switch is a type of RNA regulator that is structured and has multiple domains, and we'll be observing specifically a domain called the aptamer domain. This domain is one in which the ligand binds to, and once this binding takes place, we would have a specific mechanism. A conformational change would take place, which then would result in a behavioral change in the overall process of central dogma. So a little bit more background here. We have a second expression platform that is being signaled to once the initial aptamer domain is being binded to, and this acts as a sensor which provides that gene expression control. And interestingly enough, the ribose switch that we'll be looking at is called an adenine, so it's a purine ribose switch, and purine ribose switches are the most common ribose switches in nature. This is just found in previous studies. They happen to have both transcriptional and translational regulators, and so they are highly structured and highly regulated. And for this reason, they are this subject in general is very is a huge point in science that has had previous studies done upon it. So next slide. So yeah, a little bit of background, a little bit of previous look into the previous studies that have been done. So as you can see on the left, this is a previous model of what mechanism takes place. And so this theory suggests that in the presence of an ion, magnesium ion specifically, the ribose switch will conformationally conform from a previously unactivated state into a second unactivated state in which it's now prepared and conformationally ready to accept the ligand, and the aptamer domain is now exposed per se. Notice that this initial theory does not consider the conformational mechanism to be purely a two-state one, but instead one in which it has multiple apo states. Apo being the direct meaning being that the ligand is away from the actual ribose switch itself. And so as you can see that these various kissing loop formations on the left and right will conformationally switch in the presence of the magnesium ion, and from there have the aptamer domain be exposed to then be able to allow adenine to bind to it, which would then allow for a second conformational change and result in the so-called binding state or bound state. Now, a second study that counters this, as you can see from the figure on the right, is one that details a purely two-state kinetic mechanism, one in which the off state is present, in which adenine is not present, and it's purely inactivated, and then in which another one in which adenine does bind to the binding pocket, which you can see from the center of the figure and on the right side, and this allows for a much more highly ordered binding pocket stability, and as a result, subsequent activated translation and gene expression being upregulated. So next slide. So our researchers, through XFEL, which is X-ray free electron crystallography, through the data that was procured, have established that what seems to be the most likely solution is a four-state kinetic model. And what this is it's a bit of both of the previous theories, a bit of a combination of the two. So as you can see from the figure, you have the APO1 and APO2 states. Both of these states are existent when there is no presence of the ligand that binds, the adenine ligand. However, there is an exchange between the two. And between the APO2 and the intermediate Bate state, this being a transient state, the only difference is that the ligand has now been bound to the binding pocket. And something of significance to note is that the only residues being displaced are the uracil 48 residue as well as the adenine 21 residue. So the intermediate Bate state and the APO2 state are virtually the same, with those two residues being the only major difference. Finally, you have the fourth conformational state, which is one in which gene expression has been activated, and the signal has been sent. And so this is a state that is considered highly structured and is sort of a note of finality to it. So these are the four conformational states, and we're going to go into more on the testing done that supports these states in the next few slides.
So some of the methods that our study talked about was touchdown PCR, XFEL, which is X-ray free electron laser, SFX, and that is femtosecond X-ray crystallography, NMR, nuclear magnetic resonance, and the nitrogen labeling CPMG relaxation dispersion. Since our paper is primarily focused on the NMR results, we're going to talk about that later more in detail. So what exactly is the nitrogen CPMG relaxation dispersions experiment? It allows you to improve the relaxation states of your protein molecule. And this is important because you're able to investigate the protein conformational dynamics, typically on a microsecond to millisecond time scale. And the larger the molecule is, the harder it is for you to actually get the protein to be in the relaxed states during the CPMG experiment. And for TD-PCR, the paper synthesized two complementary DNA strands of RA71, which is the nuclear aptamer domain of the adenine ribose switch. And that was made for the RNA sample preparation. Those strands were later used for TD-PCR, and some of the steps were modified. The first step heated up the protein molecule to 95 degrees Celsius for 2 minutes. And the second step, which was the TD phase, it was also heated up to 95 degrees Celsius for 30 seconds, but this time it had an annealing step, which gradually reduced the temperature from 75 to 45 degrees Celsius in 20 cycles by 1.5 degrees Celsius each cycle. And overall, there are 45 thermal cycles for touchdown PCR. This is TD PCR is important since it will increase the specificity and sensitivity of the RA71 molecule during PCR amplification. And the next slide talks about femtosecond X-ray crystallography and XFEL, which revealed a mechanistic delay by 10 seconds. And that was able to show that there was an intermediate conformation of the protein, which is IBAID, and they did so during the mix and inject approach.
Yeah, so here we have some of the NMR spectra data of the ribose switch in both the APO and bound states. So as you can see from graph A, and then in comparison graph B has the presence of 8 additional peaks, all being uracil. And so these are directly involved in the ligand binding to the binding pocket, which makes sense. The ligand being bound is adenine, and its complementary residue would be uracil. As you can see in the previous and post NMR spectra, many of the former residues don't have a major shift in ppm. So if we can go to the next slide. Yeah, so while looking at the secondary structure of the ribose switch, the major points, the uracil, once again, are the ones in the binding pocket. So these are the ones present in the bound state. And these are the ones that are having, that present with a major shift from the apo and the bound state. Of the 8 shifts from the apo to the bound, only guanine 46 is located in the binding pocket. The next slide.
Now I'm going to talk about the NMR shifts in the amino and N7 upon ligand binding. Our paper actually had other locations such as N1, N7, and N9. However, we chose to pick N7 specifically to talk about G46. As you're able to see on both of the graphs, it's an outlier. If you look on the left graph, the x-axis represents the residue number, and the y-axis is the signal that they're measuring in parts per million, and that measures the difference in the chemical shifts. The regions at the top axis of the graph, where it says P1, P2, and so on, corresponds to the residue numbers of RA71. And basically that means that those are the RNA locations of those residues. Anything above the dashed line in the graphs represents significant differences in chemical shifts. And you're able to see that in both of these graphs, G46 is the outlier because it has a large chemical shift in NMR after ligand binding, before and after ligand binding. And you guys will see our Pymo image on the next slide. So here is our animation where it just shows you the RA71 domain, and it zooms in later into the yellow residue, which is the binding pocket. And the magenta color residues are the residues of the signals that appeared after chemical binding. The blue color residues are representing large chemical shifts after ligand binding, and U22, which is yellow colored, is the ligand in the binding pocket. So all of the nucleic acids near it are within the binding pocket, and the molecules within hydrogen bonding distances are also part of it. G46 is the cyan colored one, and like we said before, it is part of the binding pocket that displays a large conformational change before and after ligand binding. So on the next slide, in figure 5A, which is the top left corner, that is the apo state, and figure 5B is the ligand bound state on the right side. And these are the results of the nitrogen labeling CPMG results, which show the Merkel to millisecond timescale conformational exchanges of RA71 in the apo and bound states. And if you look at the yellow arrows in the Pymo images, you're able to see the residues that actually have a dynamic change before the ligand binding occurs and after it occurs. So for the top left corner here, for the comparison of room temperature B factors for apo and ligand bound states of RA71, the top left graph is a qualitative view of the average B factors per residue in the crystal structures of APO1, APO2, and BADE, and they're all color coded accordingly to the graph. And if you look at the trends of those dots, you're able to see that they all follow a similar trend and they're all clustered together. So you don't exactly know the differences in between the states. That's why in the second graph in the bottom right corner, it is a qualitative data that gives you more information of RA71 in the apo and bound states. In the yellow circle region, the residues show a shift from other parts compared to the previous figure that I talked about. And that's because it takes the differences in the average B factors per residue between APO1 and BADE and the difference between APO2 and BADE. And you're able to see that there are more areas of dynamics in the circle region. And that means that there's more change when we go from APO2 to the ligand bound region than there is from APO1 to the bound region.
So finally, we have the kinetic state model in the figure being represented by different kinetic exchange rates. Overall, the add riboswitch aptamer domain was observed to have reduced conformational dynamics in the P1 duplex. So this is outside of the binding pocket. And this is notably from the numerous exchanges, the increased numerous exchanges over time. So the KEX, resulting purely in the P1 subunit, as opposed to the increased ordered conformation of the P1 after the ligand has already been bound. And so this seems to be reasonable and matches up with is consistent with the remaining data, as the B factor analysis consistently showed low and similar B factors all across the 4 conformational states for the binding pocket. So with those similar and low B factors, it's signify that there is high stability within the binding pocket, regardless of whether the ligand had been bound or not. And it's important to remember, like, that the only significance had been that those 2 or 3 residues had been shifted over. In comparison, however, the B factor analysis seemed to show, or the differing B factors in the lower residues in the previous slide seem to indicate that the structure actually had less structure in the outer P1 pocket, the P1 duplex, which signifies that there had been a conformational exchange as a result of the ligand binding, but that conformational transformation was being taken place on the outer edges as a result of the binding pocket being bound to the ligand, but not in the actual binding pocket itself. Finally, the thermal fluctuation data had been also similar for the binding pocket all across the different states, and thermal fluctuations, which indicate increased thermal fluctuations indicate a higher a disorder and a lesser conformational stability. And so with the binding pocket having similar spatial thermal fluctuations all across, there is less disorder within the binding pocket, and at the same time, in the P1 complex having a decrease in thermal fluctuations as opposed to having it be consistent all across, it is indicative of having it go from having the P1 complex go from a highly disordered state with many thermal fluctuations, and then transforming into a more structured state after the ligand had been bound in the bound state. Key facts to note in signifying these changes: the KEX, the kinetic exchange rate of the P1 subunit in the absence of the ligand. So this is when the ligand would be not present. It was shown to have a 319 exchange rate per second. And in comparison, once the ligand had been bound to the binding pocket, the conformation the conformational stability had seemed to have increased dramatically, with the exchange rate dropping significantly to 84 exchanges per second. This would conclude our main results and data.
So some of the critiques that we noticed in the paper was specifically with the nitrogen labeling CPMG results. We stated before how G46 is part of the binding pocket and it had a significant change in the chemical shifts. However, if you look at the graph, there are other residues that aren't part of the binding pocket that also had a significant change in the NMR shifts, and the paper didn't clarify on why that is the case. Another limitation is that you can't only rely on the relaxation dynamics. You would need the B factors to actually quantify your data. And you would need it because B factors are more informative regarding the flexibility and static disorder of a protein molecule. And it provides a more complete spatial information with regards to atomic motion. Whereas with NMR shifts, the amino signals may not always be detectable. And that is the end of our presentation.